So good afternoon, class. So today's lecture is based upon two topics together. That is transgenic plants and animals together, and what are their implications on medical biotechnology. So I would first, before going into transgenic plant. So one way to define a transgenic plant is that if you take any living organism and you if we insert or if we genetically manipulate that organism by supplementing an additional gene, right? So now here there are two terms. One could be cisgenic and one is transgenic. Cisgenic could be transferring of genes from between same species, humans to humans. But if the transfer is happening between two different species, human genes into plants genes into bacteria, bacterial gene into any other organisms. So that is transgenic. Now, plants have a very unique capability to undergo genetic modification because they're in the open air and their gametes could be part of environment, right? Let's say pollen, and a pollen can be dragged away under the influence of wind, bees, birds, many agents, and these can implant or drop these male gametes onto the female portion of the flower, female organ of the flower, and there can be fertilization. And so plants must be going genetic recombination since many ages. And one of the interesting evidence that suggests that genetic modification of plant is happening since ages. We can look at the example of very interesting plant that we consume, vegetable that we consume, and that is broccoli. So broccoli, it's such a rich in so many micronutrients, minerals, and fibers. But this plant came into existence long back because of genetically modifications in the environment, breeding, and this happened in Mediterranean area. So from Mediterranean area, it moved to Europe, and then from Europe to various part of the world. So, the richness of broccoli in nutrient, its nutrient capacity for many other, for salad, for fiber. This is an excellent source. So it's a genetically modified plant in nature. So if nature is facilitating in plants such recombination or genetically modification, then if scientists are doing, then what's the problem? It's just a thought I'm sharing with you. So, so the bigger question is genetically modified plants healthy or not healthy? So there are many times it happens during the course of scientific development that we go through these developments as a wave crest of time, and we don't have a particular good answer at a given point of time. For example, people can argue that, let's say, introduction of 5G in the cell phone world. People say that 5G could be very harmful and rays and frequency, and we don't know, but we all are using it, right? And so 1G came, it went away, 2G came, 3G came, 4G came. So we are riding the crest of of technology without having a right information on human health. So this is a very interesting topic, not only from a scientific point of view, but more than scientific, you sort of understand where science could be going, right? Of course, we need to know development, what is happening, what we are consuming as a consumer. So whenever there's a debate on a scientific development, on a scientific policy, there's always a question: how does it affect us, or how does it affect me? So, for example, let's say I mentioned to you that in California they are thinking about introducing a genetically modified mosquito that is called as Aedes aegyptis. This Aedes aegyptis is aegyptis, if I'm pronouncing it correctly. This is a vector for carrying various viruses, I believe, dengue, Zika, Aedes. So they're trying to modify this mosquito so that it can dominate. So this genetically modified is benign. The genetically modified Aedes aegyptis is benign. It cannot carry the viruses, and it will dominate over those vectors, those mosquitoes which are carrying these viruses, or viruses or parasite. So the bigger question, yes, the intentions uh, could be right, but we don't know in long term what could be the consequence. And one of the major consequence is that, if you look at it from a common sense point of view, there's a chart, there is a hierarchy. For example, there is an insect. Let's say there is a uh, butterfly that feeds on plant, and yes, plants could face threat from butterfly, and one can get rid of that butterfly. But that absence of that butterfly can shift the ecological path of food wave. So if one organism is missing, then the other organism that preys on, that takes as a food, preys on that organism, it will increase in number. It will increase tremendously, and that will disturb the balance in the ecosphere or biosphere. So yes, genetically modified organisms or plants, they're important to treat various diseases. But are we going to genetically modify human beings right from the day zero, when a zygote is formed on day zero after fertilization? To what extent we are going? So with scientific development, we have to also understand the consequences of that, and especially in today's world. So let me just show you one of the slides that would convey what I mean. How important this genetically modification or so there is there's a muscular disease. This muscular disease is mainly directed by a gene or a protein. Just a second. Right? So this is a protein, myostatin. If this protein is expressed, means if the gene is active, it can stop the muscle growth, right? And this muscle, reduced muscle growth during the course of development. Muscle is important for supporting our infrastructure, our body's infrastructure to grow against the gravity. Is by our skeletal system and muscular system. And muscles, they support our skeletons, bones. So if muscles are not growing, there's a lot of functions one cannot perform as an infant grows, right? So it leads to a disease. It leads to dystrophic muscles. It could lead to various severe consequences. Uh, and so So there is another gene that can make protein follistatin. Follistatin can reduce the expression of myostatin, and then once the function of myostatin is neutralized or preempted, then muscle will grow and it will be normal. So let's say in a disease with muscular dystrophy, the 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 life could be very challenging, number one, and plus it could be very short. So 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 making of these genes which can reduce morbidity, mortality is important for humans, right? So we cannot underestimate the power of transgenic and transgenic, or in this case gene therapy, right? So I gave an example of broccoli which came out because of natural breeding, and I gave an example of very severe disease, muscular dystrophy. So yes, we need to explore, science need to develop, but again the question comes, how far, right? Uh, so one of the argument for how far is that, let's say in a lab we create a plant which has very high, which produces high level of vitamin A. Vitamin A is important for eyesight, right? Vitamin A is present in carrots and all the green vegetables, and so and our body takes up a threshold level of vitamin A. But let's say somebody has deficiency. So let's make a plant or a banana with high level of vitamin A. So that one doesn't have to take pill, just to eat the fruit and you get them. Let's say this gene mutate, because this is an artificial gene. What if vitamin A genes mutate and it can create some other kind of complication? So, by the time, so we study about the genetic development, recombinant DNA technology, but we have to understand the consequences also. So, uh, so plants are very unique organism in many ways. So plants were those organisms, those living organisms that for the first time moved from water to land. So let's say there were blue-green algae that, because of certain changes, they could propagate themselves on land, not on water, and at some point those algae, there were changes, structural and genetic changes that helped the plants to grow against the gravity. And plants are primary producer because they can take solar energy and convert into food. So, but remember that plants are those organisms, they are the first chemists and they are the first biochemists. They they in their leaf, in their body, they make so much of chemical reactions, uh, which is on which there are secondary organisms survive. So plants they have very unique property to grow independently, make their food independently, and science has developed so much that you can take a piece of leaf from a plant and you can grow the whole plant, tissue culture or propagation. So plants are not like humans that there has to always be a sexual fertilization, but plants could be vegetatively propagated by certain techniques, especially tissue culture. So, so plants inherently are totipotent, means you can take a part of the plant. For example, let's say you go to a friend's home and you in the living room, you see that plant. That plant is popularly called as money plant that grow inside living room that people keep for internal decoration for various reasons, and they don't require too much of sunlight. They can grow only with water. So you can pluck you can pluck a branch and you can bring it to your home, and then it will start growing. So plants have this kind of totipotent ability. They can start growing. Yeah. So it could be a portion of the plant. It could be a cell of a plant. It could be a tissue of tissues from any plant. So plants have that ability, and that makes it easier to create transgenic plant, right? But here, in terms of biotechnology, so what are the current problem? The current problem that world faces is rising food costs, and one of the reason for the rising food cost is low productivity, and there could be several reasons. So you can draw a concept map. Increasing price because of less productivity. Less productivity could be because the farmers who are generating this food, this agriculture, whose profession is agriculture, they are maybe dependent upon weather, right? Maybe the crop did not grow well. Maybe there was change in weather. So so low productivity, increased cost, and then people don't get. So so you need alternative route, and to grow a plant that can defy the pressure from environmental changes, or threat from a weed or from insect or from fungus. So they're more robust, more competent. And if there is increase in productivity, then there is possibility that price could go down. But is the price going down, right? We see that, okay, the prices are always going up. So where is this hypothesis that genetically modified plant will reduce the price, right? 90% of what we consume from green plants from grocery store is genetically modified, right? And the second argument that comes, that is genetically modified plants are safe or harmful. So nobody knows the right answer, but so far, it looks to be safe. But people are proposing that it could create allergy. It can create a super plant. It can totally, go in a different directions. So, so those things, uh, even if we are having some disadvantage, we don't know yet what is happening if we are consuming plants which are genetically modified. But to counter that argument, plants are in open air. They could be crossbreeding, self-breeding, breeding between two different species. So we cannot control that. So plants are one of the models that suggests that genetically modification is okay as far as plants are concerned. So major. So think about it. A farmer that grows a plant, plants the seed, inside the earth, try to water it, you know, periodically. So much of investment, and then there is a viral epidemic, right? And this plants viruses, they generally are, because it's very difficult to attack the bark, the stem of the plant. The tissue is very thick, but these viruses, they attack the leaf. The leaf could become yellow. They can become, you know um, there could be holes in the leaf. uh, these viruses or bacteria can attack the root. That will cut the connection between root and the soil nutrient, and the whole plant can die. And this could be epidemic. The whole field could be destroyed. So then you need herbicides. You need antiviral. So these viral or herbicides should not impact plant in an adverse way. So then delay of aging in a plant, delay. So let the plant stay young for longer time so that it can produce food or crops for the longer time. And of course, climate and the change of. So, so agriculture is a profession which is going every year, every season, right? And soil have limited capacity to provide nutrients, right? So, so the current soil, the current agricultural soil may not stay as healthy as what it is now, 50 years from now, because it has been used and overused and overused. So, replenishing the soil, so you need those kind of plants that can do multiple purposes. So improve the quality of seeds, improve the quality of soil, as well as make this plant more strong, right? So that's the purpose of plant biotechnology, or plant, creating transgenic plants, for increasing the productivity, reducing the price, make these plants more suitable for changing climate. So, but as far as science is concerned, science or biomedical, not biotechnology or plant biotechnologists, they take advantage of again coming back to bacteria or viruses. So there is a bacteria called as uh, agro, Agrobacterium tumoriferans. Just a second. So there's a bacteria called, as I mentioned this bacteria before, that this bacteria, Agrobacterium tumoriferans, they infect at the junction of earth and root, and the root at this junction, or root in the big in the most deep, they're very fresh, soft. So these bacteria can infect these roots, and they have capacity to grow tumor-like structure on the plant at the junction of soil and plant. And this tumor can cut off the nutrient supply from the soil, and the plant can die. But this bacteria, scientists found out that there is a plasmid called as T plasmid. This T plasmid has tumor-inducible gene, right? So any gene that can cause a pathology, a disease, it is called as virulent gene. So this virulent gene in plant is called as VIR. VIR means virulence gene. Right? So from these plasmid, these genes, the disease-causing genes can be removed, and that can be replaced by the genes which will help the plant. Right? So again, what is the strategy? Strategy is to create a plasmid where the VIR genes or virulence gene have been removed, and that has been replaced by those genes which will increase the nutrient of the plant, or make plant more resistant to herbicide, viral infection, or bacterial infection. Now, all these cloning of genes which will make plant transgenic, all these steps will happen in bacteria. So these plasmid, I'm sorry, these plasmids are binary. They can grow in bacteria as well as they can grow in plants. If you do all the cloning work in bacteria, but you don't have the gene that will help this this plasmid to propagate in plants, the plants will lose this plasmid. So this plasmid has to be selectively, selected. We have to select the bacteria which has this transgenic plasmid, and then once the cloning is successful, then this bacteria will be transformed or transfected into plant cells and selected. So dual plasmid that can grow in bacteria as well as in plants. And then these plasmids will express your gene that can improve the quality of the plant. And one of them, as I mentioned to you, that these plants which are resistant to butterfly, or these caterpillars, butterfly has a life cycle. It generally the life of the butterfly starts on the leaf, and these butterfly caterpillar, they feed themselves on the leaf. On the other hand, the leaf can get damaged. Leaves can get damaged by this caterpillar, and so scientists are developing leaves which are resistant to these caterpillars. So what is this? What will happen? That these caterpillars will die, and it is possible that whole layer of butterfly can vanish from our planet, and it is happening. So, so that will disturb the ecosystem, because these butterflies are fed by birds or other organisms that feed on butterfly. So those organisms will suffer, because if butterfly is wiped out from the planet Earth. Farmers, they argue that why to generate transgenic plants? It is, we don't know the consequence yet. There could be a totally new category of plant that could be very aggressive and take over the natural plants. It could happen. The genes could change. A gene could mutate. It is possible that a particular component is overexpressed, then it can cause problem. Uh, so there are various arguments from both the sides, and as a student of biotechnology, you need to know the argument on both the sides of this transgenic approach. So one approach of in plants is to develop this transgenic or recombinant TI plasmid, and then shuttle this plasmid, or put this plasmid in the plant that we want to modify. But in plant, as I mentioned to you, the greatest advantage is that you can take a portion of the plant, and then you can grow it in a special condition, and there is no need to do recombinant. Or you can do one time and it can produce a clone, because plants can micropropagate. We don't need to do recombination all the time, right? So what is micropropagation? Micropropagation is a practice of rapidly multiplying stock plant material to produce many progenies of plants using modern plant tissue culture methods. So, so this is not available with animals as compared to plants. Micropropagation. It could be very expensive. It is not easy to maintain a lab. You need big labs, for plant tissue cultures to grow. It could be very labor intensive. It requires more manpower. It could be expensive, and so. And if there is an infection in one plant, it can spread, and it can make business in loss if there's an infection that cannot be controlled. Right. So whenever there is a transgenic plants or animal is being designed or developed, then most of the genetic engineering experiments takes place in bacteria. So it is very important to design, when you're making the starting point for molecular or transgenic plant that is generated by genes, foreign genes. The starting point is bacteria. And it is very important that this plasmid can grow in bacteria as well as in plants or animal cells. So they have binary capacity. Number one, to grow in bacteria, as well as second, to grow in animal cell, human cells, or plant cells. Then, we discussed here TI plasmids that can be used in plant, bacterial plasmid. Then there are some plasmid that can grow in animal cells or human cells. So these vectors or plasmid, they have restriction sites in which the foreign gene can be cloned. It can be grown inside the lab culture, and then it can be, used to create a transgenic animal. So again, I might have mentioned this in the earlier class. One of the vectors that is being used to create transgenic animals is a retrovirus. A retrovirus is a virus that has RNA. That's part of RNA virus. It can easily integrate into human DNA. But the problem is, again, those genes which can create disease. So those genes which can cause disease in humans, they have been spliced out by genetic engineering. That has been replaced by cDNA, healthy DNA, and this healthy DNA could increase the expression of protein or and try to either cure a disease or to make something more efficient. So a transgenic mouse. So mouse serves as an important laboratory organisms in which various experiments can be done, because one can grow colonies of mouse, and one can easily study the behavioral pattern, the impact of genes on the growth, development, and activity. So mice serves as a very important model. It may not mimic human physiology, but at least it helps to develop insights, preliminary information, the effect of a gene. So a 8-cell embryo can be uh, taken out from the mice, and this embryo can be transformed by a retrovirus. Retrovirus is most effective way to transport a gene into cells. These retrovirus can go and integrate into the chromosomes of mice, and this mice will express the protein which is part of retrovirus. So this retrovirus vector has a gene inserted into it, and this is transformed into this embryo. The embryo is again put back into mice body, and then you see that out of the experiment may not be 100% efficient. It's possible that out of 3 mice in which the embryo cells were delivered, only 1 of them got. So these experiments are extremely tricky and tough and difficult to predict which mouse will be transgenic. So a transgenic mouse can be studied for its phenotypic effect that one can watch by naked eye, its behavior, its eating habits, its sleeping habits, and one can compare. So a lot of time mice is now being used to study anxiety, blood-brain barriers, right? Uh, so there are many medicines that are given to Alzheimer's patient and patients with Parkinson's, and it is important that these medicines cross the blood and brain barrier. So all these experiments cannot be conducted on human, but these can be conducted in mice, right? By creating transgenic mice. So in this slide, it shows that when an egg is fertilized, very recently fertilized egg will have a nucleus from a mother and a nucleus from male mice, which is called as pronucleus. And these nucleus, before they become fused, scientists can inject a gene into the male nucleus and then again create a transgenic mice. Through this process is called as microinjection. So all these things are possible. Extremely delicate, extremely high risk experiments. But mice, that's why mice serves as a very important organism tool to understand the impact of a gene. I will give an example. Again, the third approach could be take out the embryonic stem cells modify the gene through transgenic transformation, and then put back the transformed cells, mice embryonic stem cells in the blastocyst stage, and then the mice will develop. So there are three strategies that I just mentioned to you about. One is that directly insert the retrovirus with foreign gene into the embryo. That's the first approach. The second approach is to, inject the foreign gene, transgene, into the nucleus, right at the stage of after fertilization. And the third stage is stem cells. Three approach: directly into the embryo, then microinjection, and then stem cells. Now let me give an example to convince you how important this mice experiment is. So there is a uh, so you know amyloid precursor protein. When it deposits in the human brain, it can cause Alzheimer's. And Alzheimer has various symptoms, and one of the important symptom is growing forgetfulness. Right? We don't know the starting point, and so it's only people realize when it's advanced, when people start, forgetting things. So it's very difficult to work with brain stem cells and all the other approaches. So scientists have found a protein which is important for neuroplasticity, synapse formation, and iron export. This is a 4-kD protein. So an amino acids, 39 to 42 amino acid is derived from uh, this amyloid precursor protein. So APP 717 contains phenylalanine instead of valine. So there is a change here. APP 670 to 671 contains asparagine and leucine instead of lysine and methionine, right? So these effect of these mutations cannot be studied on human brain, but these two proteins can be easily expressed in mice through three technology that I expressed to you, either directly transformation of embryo, or through stem cells, or through microinjection. And then one can study the pathology of these protein, this gene. So amyloid precursor protein, this can create plaques in human brain. That could be, there are various theories for the pathogenesis of Alzheimer. One theory is that there are a lot of proteins that deposits in the brain, and once it starts depositing, it keeps increasing. And there are many hypotheses for that. So this is one of the approach. Right? So So, right. So I would like to stop here and take your questions if you have. And if you don't have any question, then I would just pause the recording